Hello, my name is Jacob Levenstein, and I'm a PhD student working with Professor Charlotte Stagg at the University of Oxford and Dr. Peter Bandettini at the National Institutes of Health. Today I'm excited to share with you our results looking at multi-voxel spectroscopic imaging during periods of rest and task. Specifically, we're interested in looking across the motor cortex at neurochemical changes in GABA and glutamate while participants perform motor tasks. To provide some background to this work, I'll begin with the technique that we're not using, but is commonly used. It's referred to as magnetic resonance spectroscopy, and it allows a user within a single volume of interest to collect information about metabolites and their concentrations. Here, depicted in purple, would be an example of a single volume, and these are traditionally collected at rest. On the right is the technique that we are using, and it's known as magnetic resonance spectroscopic imaging. Here across the brain in red-yellow, I'm showing you the spatial coverage of where we collect 119 separate voxels, and I'm displaying to the right 6 such voxels, and if you zoom in you can see that the black line represents the raw spectra, the red line is the basis set model fit, and below that are the residuals. Using a traditional MRS approach with a 2 centimeter isotropic voxel, we now with MRSI can represent that single voxel with about 21 independent voxels. This allows for greater spatial investigation into neurochemical concentrations across the brain. Now, using this increased spatial specificity of MRSI, we wanted to ask how neurochemical concentrations change across the cortices when performing a motor task. Specifically, we are interested in looking at concentrations of glutamate, which is a major excitatory neurotransmitter and also plays an important role in energetics. And we wanted to see if during our motor task we would see task relevant, meaning sensory motor areas, showing increases in glutamate. Conversely, we are also interested in GABA concentrations during our motor task, as GABA is the major inhibitory neurotransmitter of the brain. And we hypothesized that GABA would increase, meaning inhibit, areas outside the sensory motor region that we're anticipating glutamate will increase. We also hypothesized that local areas will show GABA decreases, which would be disinhibition, which may facilitate our task. Now, those hypotheses aside, understanding where neurochemicals are changing in response to a task is a relatively new area and not much is known. The study that I'm presenting today involved 15 healthy participants completing a single session involving a resting MRSI scan, where participants had their eyes open and were instructed to maintain a soft gaze on a fixation cross, and an MRSI active task. In this task, participants used in their right hand a force transducer, and they were able to control a green ball, causing it to increase as they squeeze and decrease as they relaxed. Now, the 50% line, where they were instructed during green trials to squeeze and get the ball to rise to, was scaled to 50% of their max voluntary contraction. Both the rest trials and the task trials were on the screen for a second each, making this a 0.5 Hz continuous motor task. The first result I'm showing here involves where spatially we were able to collect GABA and glutamate after post-processing across all of the participants. The frequency plots indicate the number of participants who had that particular voxel involved within the study. So to say this another way, the voxel intensity at each point represents the number of participants who had that particular MRSI voxel included following post-processing. The next result involves ROI-based analyses, looking at both the left and right precentral gyrus. Focusing on one panel at a time, this first row is indicating that there's baseline symmetry between the left and right precentral gyrus concentrations across the participants for both GABA on the top line and glutamate on the bottom. In this next panel, we're now looking at the asymmetry during the task MRSI. And here, we no longer have a relationship between the left and right precentral gyrus. This is not surprising given that we have a unimanual task, but it does indicate that both hemispheres are reacting to the task in a different way than we had seen in the baseline symmetry to the left. The next question we wanted to ask is whether or not we observed changes in the left hemisphere from baseline to task. And for both GABA and glutamate, unfortunately, we do not see a consistent pattern of increases or decreases within our precentral gyrus left. This is also true when we observe in the right precentral gyrus. Again, we do not see any difference across participants where they're consistently changing in one direction or the other. To summarize these ROI-based analyses, we find baseline symmetry between the left and right precentral gyrus for both GABA and glutamate. During a task, these are disrupted and we no longer find that relationship. The rate of change in terms of baseline to task was not consistent across participants within the left precentral gyrus for GABA or glutamate or within the right for GABA or glutamate. After observing that participants move in different directions given our task, we wanted to know whether or not baseline concentration had any prediction power on the amount of change during the task. In the bottom left panel, we observed that glutamate concentrations at baseline were strong predictors of the task change. Here, we can also observe the number of participants who had increased glutamate during the task within the precentral gyrus left and right, as well as those who had greater during the baseline. In the bottom right corner, we can evaluate this relationship using the GABA concentrations, and we find that for the left precentral gyrus, which was the task active hemisphere, there is a relationship of baseline concentrations predicting the amount of task change. However, in the right precentral gyrus, we do not observe this relationship. To summarize the analyses investigating baseline concentrations predicting task change, we observe this in glutamate for both the left and right precentral gyrus, and in GABA for the left precentral gyrus, with right precentral gyrus approaching significance. Moving away from the ROI-based approaches, next I'm going to show you the heterogeneity in neurochemical responses that we observed. So to orient you to the color bars here, in red-yellow is where you'll see neurochemicals that increased from baseline, whereas in blue-light blue, this is where the concentration was greater during the baseline resting scan than during the task. Focusing in on a single participant and looking specifically at their task active hemisphere, within glutamate we can observe local excitation on the posterior bank of the precentral gyrus and to a greater extent on the anterior bank of the postcentral gyrus. We see an inverse pattern in their GABA where we see this local disinhibition. We also observe areas of greater decrease in glutamate and increase in GABA on the anterior side of the precentral gyrus. In a second participant, we again observe areas of local excitation where glutamate was greater during task than rest, and the inverse pattern in their GABA where GABA was greater during the baseline than it was during the task. That said, we also observe in this participant again areas where glutamate was lower during the task than during rest, and the inverse where GABA had increased inhibition during the task. Unlike in the first two participants where we observe local increases and decreases, in this third participant we see widespread glutamate increases, and conversely we see widespread GABA decreases during the task. Taken together, it's clear that with this particular task, the spatial profile of neurochemical change is heterogeneous across participants. To conclude, I'd first like to say that the novel use of magnetic resonance spectroscopic imaging used here during periods of task and rest allow for an investigation into how neurochemicals change across space. Using this technique, we first observe symmetry between concentrations of GABA and glutamate relative to the left and right precentral gyrus. This relationship did not hold true once participants performed a task, and this is not necessarily surprising. What is surprising is our next results, which showed that the direction of change within the precentral gyrus was not consistent across participants. We follow this up and find that baseline concentrations were predictive of how much change we would observe while participants performed the task. We find a significant relationship between glutamate change in the left and right precentral gyrus and GABA change in the left precentral gyrus, but not the right. Finally, I show 3 examples of spatial heterogeneity in terms of how neurochemicals change relative to this particular task. In light of these findings, future studies interested in task-induced neurochemical change, whether across space or time, should consider the heterogeneity across participants observed here. With that, I'd like to thank you for stopping by the poster and encourage you to get in touch if you have any questions, comments, or would like to discuss this work any further. Thank you.